So a lot of the work that I do, I'm a gut physiologist. My interest is primarily looking at the mechanisms by which food is disassembled, so matrix effects, how this affects nutrient bioavailability and then metabolic responses. Peter Ellis and I work primarily on food structure, particularly chickpeas, including hummus. And we're interested in this, one, from a sustainability point of view. Nobody eats enough pulses in the diet. They're easy to grow. It's good for diversification of agriculture. And they're an exceptional source of dietary fibre. So as a gut physiologist, my interests are primarily just looking at what happens in the gut. I'm not going to talk about the microbiome. I only work on the small intestine, or the upper gut. And in terms of the food that we eat, my interests are the physical properties of the food, particle size, the particle structure, the chemical structure, and the influences of other food components. How this affects biochemical and biophysical processes. So we've already heard a lot about viscosity, how this can affect enzyme kinetics, which is primarily what I'm going to talk about today, and nutrient diffusion. And then the physiological outcomes, how this affects motility in the gut, rates of digestion, and then the rates of absorption, and then ultimately the altered metabolic response. Actually, if you were here at the last meeting, I gave exactly the same slide. And again, as Marianne talked about, dietary fibre, we're primarily talking about the components of plant cell walls. Cellulose, hemicelluloses, pectic substances, and then the non-carbohydrate components. There are important functions for the plant, including structural support, and then a barrier for the external environment. I'm going to skip over this quite quickly because everybody's already talked about it. What I will highlight, though, is the way we consider dietary fibre from a nutritional perspective and how we've classified it from an umbrella term that incorporates all of these things to trying to redefine it to include things like soluble fibres versus insoluble fibres. From my perspective as a basic scientist, I like to consider all of these chemical structures independently, but I am somewhat guilty of trying to group things together into different categories, as we've done in this review. So what I'm going to talk about today is the mechanisms by which fibre might act in the gut and how this can affect glycemia. So obviously the first is the rheological properties, particularly the non-starch polysaccharides, such as oat, mixed linkage glycans, legume galactomannan, which is one of Peter Ellis's favourites, and we'll talk a bit more about that today. But also the importance of hydration and particularly molecular weight, which is extremely important in terms of whether you're actually going to see a physiological function. So this is an example where galactomannans were fed to piggies, and they looked at the change in, as you increase the viscosity in the lumen, you see a decrease in plasma glucose. There are other interactions, including molecular interactions between fibres and substrates like starch, enzymes like alpha amylase, or endogenous components like the mucus layer that can affect diffusion, absorption, and so forth. And then there are intrinsic structures in terms of the matrix of the plants. So encapsulation, if you can maintain the structure of the cell walls. Marianne already talked about that in wheat. Last time I spoke about this in terms of our novel chickpea powder, which I'll talk about a bit more, and how this can significantly regulate rates of digestion and absorption. So Peter Ellis's favourite molecular structures, so galactomannans from guar gum. Guar gum has a lot of interest in the media at the moment. A lot of people looking at how these emulsifiers can potentially have deleterious effects, and they're synthetic, but guar, galactomannan, is a natural product, comes from cluster beans. As you can see here, it is a beta D14 mannan backbone with galactose side chains, and as a guar flower it contains about 82% galactomannan. It's water soluble at room temperature and forms a viscous solution at quite low concentrations. Or obviously this depends not just the temperature but the molecular weight. The higher molecular weight, the higher the viscosity at lower concentrations, but how you get it into solution becomes more difficult as the molecular weight gets larger. And on to my interest, which is Pulson. I should probably declare a conflict of interest here in the fact that I am an inventor on some of the patents associated with this. But this is where we have a hydrothermal process which you can cook a chickpea, you can separate all of the cells, and the cell walls around the individual cells remains completely intact, entrapping all of the starch and protein within. The idea being that this can slow down or limit enzyme accessibility to its substrate, reduce the rates of starch hydrolysis, and potentially reduce the product from leaving the cells and affecting rates of enzyme activity. Levels of hydration, the processing conditions, how you've prepared everything has an influence on how this can affect rates of hydrolysis. And we've done some work now looking at how you can incorporate this into a food matrix. So this is a confocal micrograph of what the individual cells look like. So in green is the starch, and the blue is the cell wall material that surrounds it. And we've been able to bake this into breads. And as I talked about in a last meeting, you can have a significant reduction on plasma glucose postprandial. And experimentally, when we put some labelled enzyme into a solution, we can measure the rate at which the enzyme can enter the cells. And it's significantly reduced as long as the cells remain intact. So we did publish all of this. We demonstrated that including this pulse fibre into bread rolls at 30 and 60%, we saw significant reductions in the plasma glucose response, particularly early on. However, interestingly, we only saw a significant reduction in plasma insulin at the very high dose of 60%. Now, that's great from an experimental perspective, but eating a bread roll that's made out of 60% chickpea cells was tough for our volunteers, shall we say. So this is what led Peter and I to the idea that is there potential for looking at synergistic effects of different fibres with different chemical and physiological mechanisms. So mostly what I talk about today is what happens when you combine fibre with different physicochemical properties. So our intact cell walls from our pulse-on chickpea powder, and a viscous soluble non-starch polysaccharides of the guar galactomannan. And will combining the fibres with the different mechanisms have a synergistic effect on postprandial glycemia. So we did this by doing in vitro analysis using single enzyme assays and the harmonised Infogest digestion assay, and then we did a postprandial study in healthy humans. The breads that we made were based on white wheat flour with a 30% substitution of the pulse-on which is the lower dose that we used in the previous study, but we included 8% of the guar galactomannan. So the three bread types we analysed in this part, so for the human study was the wheat bread control. We had an isoglycidic bread, which was matched for the available carbohydrates in the control, but with the 30% chickpeas and 8% guar. And then one that was isomass, so just to see what happens when you feed something that's the same total mass for the meal of the wheat bread control, but had significantly lower levels of available carbohydrate. We did a single enzyme assay using pancreatic alpha amylase, and we fixed the ratio, the ratio of enzyme and substrate. So when you look at the digestion curves, you can see that the inclusion of the pulson and guar significantly reduced the rate at which starch hydrolysis occurs. And following this, we can then analyse this data a little bit more to look at the rates at which the reactions are occurring using the logarithm of the slope analysis. I'm not the enzymologist, we had some help doing this, so please don't ask me questions. So for the wheat flour control, where all of the starches come from a single source, we can see that, oops, I've broken it. We can see that you get a single slope, so you're getting a single rate of reaction occurring throughout the digestion. But with the incorporation of the guar and the pulson, you see a split, you see a two-phase rates of digestion. So we would hypothesize that the first fast rate is occurring as the breakdown of the starch from the wheat flour, and then you're getting a slower rate from the starch that's from the pulson. Interestingly, the rates are somewhat different between the isoglycidic and the isomass, despite the fact that the composition of the bread is identical, and we think that this is related to the differences in the substrate to enzyme ratios. When we look at a more complex in vitro digestion, so the infogest, so this is a standard three-phase digestion model, where you look at the oral phase with salivary alpha amylase, the gastric phase with pepsin lipase and a lower pH, and then the intestinal phase where we use a pancreatic extract, but it contains pancreatic lipase, pancreatic amylase, and proteases. We can try and simulate what would happen during the entire upper gut. And you can see, again, there is a significant difference between the wheat bread control and those that contain the pulson and guar. Early on, we can see that there's a difference between in the stomach and the oral phase, and this comes down to sort of the macrostructure of the bread, that as you break it down, the guar and the chickpea change the rate at which or how it breaks down, so you get different particle size in the bolus. That's going to slow rates of digestion. And then the intestinal phase, we're looking more at the microstructure, the chickpea cell walls, and the influence of the guar in terms of viscosity. So when we fed these bread rolls to some humans, we did a postprandial study where they were fasted overnight, came in, ate a meal with 50 grams of available starch, carbohydrate, and measured the plasma glucose and insulin over a 4-hour period. So for the glucose, we did it in two methods. All the participants were given Abbott Freestyle Libre continuous glucose monitors. This is a good way of telling whether participants actually fasted before they came in, because we could measure beforehand. And second, it's a slightly different measure as you're measuring interstitial fluid rather than venous blood. And what was interesting there is that while we saw a significant decrease early on, this relates to the decrease in available starch from the wheat flour, but at a later time point, particularly for the isoglycidic bread, you start to see a second increase in plasma glucose. What was interesting is we only saw this in the continuous glucose, that was not, we did not see that in the plasma samples, partially due to sampling time, but I think partially due to the fact that we've lost that because the glucose has been utilised for metabolism. For insulin, again, we saw a significant reduction in plasma insulin over the postprandial period. Importantly, we saw it both, we saw it here with a 30% pulse on, whereas we didn't with the original study that had no GUA. So GUA, galactomannan, is a significant contribution to looking at to the insulin response. We see a significant insulin sparing here, and we see that too with the C-peptide. In terms of satiety, as you might expect, the isoglycidic bread, which was significantly larger and greater mass for the meal, because the fibre results in a higher moisture content, showed a significant increase in satiety. But again, we think that this is just a mass effect of the total meal rather than anything specific. What's quite interesting is that for the hedonic scores, where we asked the participants about what they felt about the consumption of the meal, we saw no significant effects of aftertaste, texture, or taste and enjoyment. That's important for the chickpea because the beanie flavour that you normally get with a lot of legume products was not a particular problem in this case. We saw reductions in the dryness. Obviously, with the increase in fibre, there's more moisture in the bread. There was some difficulty eating the ICB and the isoglycidic bread, but that's again because it was such a larger meal. Importantly, compared to the previous study, we found that including the galactomannan had significant benefits in terms of the texture of the bread, so we could improve the quality of the bread as well as any kind of nutritional products. So to conclude, as an umbrella term, dietary fibre encompasses compounds with diverse physicochemical properties and varied physiological benefits, which everybody has discussed already. The combination of fibres with complementary physiological mechanisms can be a good way to design foods with enhanced nutritional properties. So, and this is where we like the idea where you're combining the chickpeas, where you're looking at a physical barrier to the guar, which is a viscous fibre. The effects on satiety are equivocal, but this is a short-term study. But ultimately, we would like to see that there's a greater understanding in food structure and how this can influence physiological aspects. I was also going to talk about something else, but I will leave it there. Thank you.